are tacos, one per person.
Can we just get rid of this?
So I try to hold it straight up.
It's probably what it is. Cycling technology of the future. Just like gambling.
Chairs here. Should we put some? Oh, this is fun. I know. Wait a couple minutes. Good morning, Dr. B. She's all set. But if you it would be nice to have. Yeah, sounds good. So you'll stay the same. You'll do the same.
All right, thanks so much. Welcome, everyone. I'm John Cook, Chair of the Department of Cardiovascular Sciences, and we're going to have some exciting new information for you on our RNA therapeutics program. And I'll let you go ahead and make the introduction, Dr. Bruno.
As many of us know that Dr. Bruno is a great scientist and director of the clinical RNA core. She was trained in a broad aspect of scientific field and has lots of industry experience. If we look at her academic training, she obtained her master and Ph.D. In the medical microbiology, biomedical sciences, human genetics. And she got her 2 postdoc training in MD Anderson and Baylor, which was in the field of human molecular genetics, RNA biology, mRNA regulation. In industry, she developed microbiogenetic testing platforms, non-viral gene therapies, cell-based immunotherapies. And in Generation company, she led scientific teams successfully applied stem cells as a source of regenerative medicine for various indications. Dr. Bruno optimized standard operating procedures for the cGMP manufacturing of T-cell immunotherapies for clinical trial phase 1 and 2 studies in the United States and Europe for those patients with hematopoietic malignancies undergoing stem cell transplantations. Dr. Bruno is currently leading the development of translational approaches for the advancement of mRNA-based therapeutics, which provide safer and more efficient regulation of gene expression in cell-based regenerative and immunotherapy treatments. Today, we'll share with her work in progress that's next generation therapeutics, RNA drugs for the treatment of senescence and inflammation.
Thank you so much for that introduction. And good morning. Thank you for coming here this early and for being part of such an energetic center. We have an amazing story to tell. Actually, today I'll be telling two of our mainstream stories that we are working on, and this is a team effort. The work that I'm going to present is members of the RNA Corps have contributed significantly to the progress that I'm about to show you. So today I'm going to tell you the story of what happens when you're trying to achieve or use somatic cell therapies for any therapeutic indication, including cancer, regenerative medicine. What happens to the cell? What's some of the concerns and some of the challenges that the field faces today? So when you're trying to use somatic cell therapies, you have to take these cells, sometimes genetically modify them, sometimes ex vivo expand them. And that leads to a series of challenges. One of them being, can you generate enough cell product to achieve a therapeutic benefit? The aging of the cell as you expand them ex vivo, and can they expand in sufficient numbers in vivo and maintain a proliferative capacity? And their persistence also in vivo is critical for achieving a therapeutic benefit. And in addition to that, a lot of these cell therapies rely on genetic modifications, so the use of DNA or RNA molecules in order to achieve that and change gene expression. And one of the main challenges are how do you achieve a highly effective method of nucleic acid delivery in the absence of viral approaches. Today, the story I'm going to tell you relates to a type of immunotherapy that we call adopted T-cell transfer immunotherapy. What happens during adopted T-cell transfer is that you either have, in the case of a autologous donor, which is the patient itself, the cells, mononucleated cells are taken from the patient. They are processed in a way that selectively the therapeutic cells are selected, in this case T cells. They're removed from the patient. Ex vivo, they are activated, they're modified. A lot of these genetic modifications are achieved through viral approaches. There are also currently some non-viral approaches that are being developed. And then once modified, they're selected, expanded, and infused back into the patient with the hopes that the cells are now more prone to recognize specific tumor antigens. This therapy has revolutionized the field of cancer therapeutics and has been recognized in all major high-impact journals. So what happens? One of the things that you do when you're trying to manufacture this type of therapies, you take, as I mentioned, you take from an apheresis product from a patient. An apheresis is a highly cellular fraction that has your mononucleated cells and specifically your immune cells. And you selectively expand these cells using appropriate culture conditions. All of this has to be done in a very controlled manner. So T cells you expect the cells to proliferate, and as you can imagine, the biological variability that you will encounter depending on who the donor is for these cells is highly significant. Then these cells, once activated, then they are, with the intention of expanding them and achieving high numbers, then they're genetically modified. In this case, a lot of the ongoing clinical studies are using viral gene transfer using viral approaches. And then you select them further expand them, and cryopreserve them. A lot of the times you need several doses in order to achieve a therapeutic benefit, and then they are infused to the patients. So what are some of these proliferation challenges that you'll face? One of them is that you need to achieve billions, a very high number of cells. Most of the time for an average individual of average weight, you will expect at least 10 to the 9 to 10 to the 10 cells for one single dose. And in addition to that, post-infusion, these cells have to continue expanding, and they need to be able to maintain the level of activity necessary for them to have their anti-tumor activities. But unfortunately, what is being observed is a lot of the times these cells are forced to expand to the point that they reach a level of senescence. So they either die, they apoptose, and they become senescent. This is a very interesting article published. It's a review published by the Rosenberg team, and he's an expert in the field of immunotherapy. And it's actually compiling data from 1994 to 2014. And here you see the use of chimeric antigen receptors, or TCR receptors, to guide the cells or modify the cells to achieve anti-tumor activity. Now, if you can see, the field is really booming. Anywhere from 2000 to 2014, there's been a significant increase in the number of protocols that have been submitted to the Recombinant DNA Advisory Committee, the committee that oversees these studies through the FDA. But still, these type of therapies, if you notice, a lot of these targets are geared toward solid tumors. In the case of hematopoietic malignancies, there's very promising results, but the solid tumors are still remain to be seen high effects of immunotherapy in this particular case. So what happens? Why is it so much of a challenge? There's a significant amount of evidence that suggests that the cells tend to senesce, as I mentioned. So senescence involves a series of events, anywhere from epigenetic modifications, loss of mitochondrial dysfunctions, and cellular senescence can involve any of these events, alterations in cell-to-cell communication. So what we are seeing, even genomic instability, bless you. What we are seeing here is that there's significant evidence supporting that one of the key events that leads to this cellular senescence is the shortening of telomeres. So this study was first reported in 2007, where they look at actual clinical data and clinical samples, and they look at the telomere length of T cells that were forced to expand and were expected to persist in vivo. And the data clearly show that a correlation between the persistence and telomere length. So those that have a longer telomere length tend to persist more. And again, this is patient data, compared to those that have shorter telomere lengths that did not persist as much. So this correlates also to the therapeutic benefits that they saw in the patients. This is data from Dr. Cook's teams, where they looked at the effect of enhancing telomere length by using an RNA molecule expressing, encoding a human telomerase. So fibroblasts that were forced to expand and proliferate significantly ex vivo, in vitro in this case, were treated at different time points with human telomerase. Now what they observed is that three initial treatments resulted in an increase in proliferative capacity in these cells when compared to a catalytic inactive control. So this suggests that indeed an increase of telomerase may allow the cells to achieve a higher replicative capacity. So what is some of the innovations that Dr. Cook and his team and the core have been able to achieve is that they've been able to achieve the first level, the first preclinical evidence that indeed H3 and the expression of human telomerase in chimeric antigen T cells containing chimeric antigen receptors could indeed result in an improved cancer immunotherapy. So the first preclinical evidence suggests, as you can see here, this is telomere length, and then you have T cells genetically modified with GFP, with just the chimeric antigen receptor, the chimeric antigen receptor in combination with inactive telomerase, or the chimeric antigen receptor containing the telomerase, encoding the telomerase as well. So you can see there's an increase, they observe an increase in telomere length. And this increase in telomere length also correlated with an increase in persistence, and they went all the way to 135 days. And these are in vivo studies in mice. And more importantly, they observed that this persistence of the cells resulted in an increased survival of cells of mice carrying multiple myeloma tumors. So what we are trying to achieve now is use these therapies, modify existing chimeric antigen receptor T cells in order to achieve an enhanced proliferative cell, so a cell that has a higher replicative capacity. One of the goals of this project is basically to develop and test an enhanced telomerase. What we're trying to do, and I'll tell you in more details how we're trying to achieve that, what we're trying to understand how we can use RNA molecules to express telomerase and have the cells have achieve a higher persistence in vivo and in vitro. In vivo studies will allow us to better assess the effects of having T cells, CAR T cells encoding in human telomerase. And we're also trying to achieve protocols or develop protocols that will allow us to do this in a manner that is in accordance with the manufacturing guidelines of the FDA. How do we achieve that? So I'm going to tell you a little bit more in details how we're trying to achieve these goals. First of all, we have RNA, it's perceived as a safer approach over DNA, and especially over DNA using viral methods. So you reduce the risk of DNA integration, and it lingers for a short period of time. So you just want RNA is very useful for an approach where you just want to provide certain cues to the cell. The RNA vectors that we are developing are simplified vectors in a sense that it has minimal sequences that can confer the expression of the RNA and the synthesis of the RNA in vitro. Now, saying simple is not really an accurate description of this, because there's some key sequences within your RNA that are very important, and they can confer higher stability to your transcript and lower immunogenicity. In order to achieve that, we have incorporated in all the RNAs that we produced flanking regions, the untranslated regions, that have elements that confer higher stability. In addition to that, we have a longer poly-A that enhances the stability of the transcripts, and we're looking in the context of the T cells, what codon and what sequences are recognized as self and what sequences can confer higher stability in a given transcript for a given cell type. So how do we achieve that? As you know, and this is just a cartoon describing what happens during DNA transcription and translation. So once the DNA is transcribed, the RNA is further processed in the nucleus, exported, and then these RNAs undergo a series of modifications in a sense. They're either recognized by protein complexes that could confer higher stability, and this is also a lot of the times is sequence-based, or they're immediately then taken into the translational machinery and they're translated, or they can be destabilized and degraded. So we need to understand, in order to develop molecules that can have a therapeutic benefit, and that can be efficiently recognized and translated, we need to understand what sequences confer a higher stability as opposed to destabilize the RNA and make it be degraded. Now, in the case of our RNAs, the way we're pursuing this, and this is work that Sahana is currently doing, she has blocked transcription. And by blocking transcription, you stop the formation of any new nascent mRNA molecules. The way she has blocked transcription is using actinomycin D. Actinomycin D is actually used also as a chemotherapy agent. What it does is it binds to the site of transcription initiation and blocks RNA polymerase from elongating. So you will no longer make any new, and that allows you to look over time at the stability of the different RNA molecules that are existing. So she has done these experiments, and what she's done is then she's looked at what happens, can they indeed, we confirm that transcripts that are known to have lower stability, indeed they start decaying and they're degraded over time, versus transcripts that are expected to have a much higher stability, they maintain that level of stability over time compared to the ones that are less stable. So now we have we have 3 different donors, blood donors, and these are primary T cells that were treated with actinomycin D. RNA was isolated, and we have sequence analysis from these RNA-seq analysis from these samples that will allow us to then understand which transcripts over time are more or less stable, and then what confers that stability. This work is going to be done in collaboration with Dr. Chen and his bioinformatics team. So next time we talk, I hope I can tell you more about, indeed, what sequences can confer higher stability. Why do we think this is possible? So the reason why we think this is possible is based on a recent report from Dr. Jeff Coller's lab at Case Western, where they look, and this was in yeast, they look at transcripts that have a much higher stability versus transcripts that have lower stabilities, again in yeast. And they observe that certain codons and the order, the position, and the type of synonymous codons that the cell preferred could confer in a given transcript. And again, this is the same transcript, a different transcript encoding the same protein. But as you change from one synonymous codon to another, you end up with a very different level of stability. Again, indicating that the sequence, indeed, which codons are used, determines what level of stability that transcript has. And also, they observe that optimal codons could also were more efficiently translated as opposed to those that did not have optimal codon sequences. What else do we observe? In addition to that, what we are trying to achieve, again, we are a very translational team. So everything we do, we're trying to make it in a way that is translatable, that we can then take it into the clinic. What we do is we make RNA molecules, and we're trying to understand not only how to, at the level of the sequence, but also at the level of how to manufacture them in a way that we have highly pure and stable molecules. So this is just schematics of our current process. We have a series of quality control points to ensure that indeed it's a control process, and to ensure that indeed it's a reproducible process. And again, we follow the guidelines, the FDA guidelines and GMP guidelines. We have several areas where we face challenges. So this is not just you go from a research bench to just a GMP facility and you're fine. There are several challenges that you have. So imagine as you upscale these processes, you also need to understand the optimal enzyme conditions. And we're in the process of optimizing our own chemistry to make these RNAs. But we also have to understand the kinetics when you have large volume enzymatic reactions. We also are trying to look into, again, that's true for the linearization of the template and the in vitro transcription of the RNA. The HPLC team, Dr. Sukovershin and Emily Lawless are working, and they've made a significant amount of progress, and I hope they get a chance to also tell you what they've done. Looking at novel ways of purifying these molecules, and in addition to that, looking at upscaled and controlled ways of achieving that. So this is a team effort to be able to achieve readiness to make these molecules. We also are looking post HPLC, there's additional steps that have to be done in a highly controlled manner. Now, just to tell you a long story in a very short time, we have data to support that the process generates impurities that could be highly immunogenic. We have been able to identify these impurities, and not only that, with the quality control team led by Andrea Balog, we have been able to develop and validate tests. In addition to that, we have been able to purify and remove these impurities using HPLC. Just to quickly show you, in the absence of any nucleotide modifications, and that again is a story for another conversation, but if you were to have unmodified RNA that is unpurified versus purified with HPLC, you significantly reduce the levels of cytokines that are released by the cells. If you add modifications that are known to confer lower immunogenicity, you even achieve a much better level of less toxicity and less immunogenicity. And this is important when you're trying to generate molecules that take you to the clinic. And not only that, you achieve a much improved level of transfection efficiency, in this case, nucleofection efficiency. So Dr. Suresh has been able to achieve a very high level. This is actually, we are the first group to achieve this level of nucleofection efficiency in T cells using highly purified HPLC RNA that Dr. Sukovershin and Emily have been able to prepare for us. This is just a list of tests that allow us to ensure that these are highly qualified molecules that can be taken into the clinic. And this is just part of it, of the list. QC team works overtime, to be honest. And I just wanted to briefly tell you, that was just an example of how we are achieving higher proliferative capacity for immunotherapy in this case. But we also are looking at non-viral methods of delivery. And I want to tell you, these are some of the molecules that we're actually, some of the particles, sorry, that we're looking into testing. And this is possible because we're embedded in a research institute that has an army of nanomedicine scientists. And we have the capability of taking what's being developed here in-house and testing it for RNA delivery. So it's a unique synergistic collaboration. And in addition to our nanomedicine scientists here at our institute, we're working with Northeastern investigators to achieve increased the delivery of RNA. Just to give you a story, and then again, my story was about inflammation. So one story where we tested these delivery molecules to achieve lower inflammation is looking at RNA, microRNAs that could regulate inflammation. If you look in the literature, you'll have a series of microRNAs that have been implicated in having a role for controlling inflammation. The reality is we couldn't find solid and concrete evidence that indeed they can do that. So we tested two microRNAs, miR-125A and 125B. The way we deliver this to the cells is using these very interesting and novel biomimetic guanine and cytosine-like molecules that have a lysine tied to them. So these bases, these biomimetic particles, actually, they self-assemble very easily. And they look almost like a tire, like a car tire. And they assemble in this structure. So they have, again, a cytosine and guanine motif linked to a lysine. They self-assemble into this ring that is called the rosette ring. And then they stack up in a way that is quite interesting. And as you can imagine, having the lysine at the end, it confers a positive charge to these particles. And they have halos, and they can be modified to be of several sizes and lengths. And this is all in collaboration with Dr. Hitchin-Feneri. He's one of the inventors of these amazing rosette nanotubes, and he's at Northeastern University. So what we did is we first look, and this again is the work done by Andrea, Carlo de los Reyes, and Enrique Guerrero. And they looked in two different cell systems. One is the macrophages, and the other one human umbilical vein endothelial cells. And they actually observed it, just to give you a quick overview here, this is just viability. The cells did not mind being subjected to treatments with these nanotubes and microRNA complexes. And that was true for both cell types. And they efficiently were able to encapsulate, and this is amazing, they were able to encapsulate not only short RNAs, and these were not very short RNAs, they were actually precursors that had an additional domain. So there were about 250 nucleotides. And they were able to efficiently encapsulate these RNAs. So as you see, this indicates the lower amount of free RNA in the sample, in the complex. So you can see as they found an optimal ratio in which most of the microRNA is complex with the RNTs. But they also achieved that with longer RNAs, with 1 kb and 2 kb RNAs. And now they have done it with 4 kb RNAs. So that's quite impressive. And these are, having the RNAs complex with these rosette nanotubes protect the RNA. So in the presence of serum, RNA will not last long at all. But you can see that the stability of the RNAs is significantly increased when they are complex with rosette nanotubes. K1 and K3 indicates the position of the lysine. And cells uptake, I hope you can see this well, but in this case, the RNA is the one labeled. And the labeled RNAs that are complex with the rosette nanotubes are able to localize to the cytoplasm highly efficiently. And to the point that when compared to lipofectamine, they're a lot more efficient. And what you see here is lipofectamine, which is what we normally use at the bench. But this is very low levels of microRNA. I think it's in the nanograms, if that's correct, nanograms levels. So you see that you don't see at the level of very low, 10 nanograms, you don't see the microRNAs. But when you use the RNTs, you can efficiently see the RNAs localizing to the cytoplasmic region of the cells. And then we looked at, okay, so now that we have a system in which we can deliver, can we reduce inflammation? What are the roles, the functional roles of this RNA? So having the right delivery vehicle allows us to look at inflammation. So macrophages, this is looking at nitric oxide production of macrophages. And you can see here when you use an antisense of the microRNA, so we are knocking down the microRNAs, you have an increase in inflammation. This is very interesting, and it's quite significant when you have the sense, and this is a mistake here, a sense microRNA, you have a reduction of in inflammation when delivered with these particles. So this was just a little bit of data, because we have many projects that we have ongoing in collaboration with nanomedicine scientists and in our team that are actually looking at delivery vehicles. So this particular project will look now into biodistribution in vivo of these rosette nanoparticles, and again looking more closely into inflammatory responses when you deliver these microRNAs. And we will continue to look into manufacturing these large or small RNAs in a manner compliant with the FDA. I really appreciate your time and your paying attention in that. And I have to acknowledge that this is work from an amazing team. I would say we're like family. The RNA core, an amazing team that has been able to achieve all of that. And we have the support of the GMP team. And then, as I mentioned, an army of nanotechnology, excellent nanotechnology scientists that help us achieve these advance these technologies. So thank you so much, and if you have any questions, I'll be happy to.
Any questions?
Just a very short question. It's very interesting for the growth set nanotube. So you mentioned is how efficiently it can capture RNA. But how about the release kinetics?
The what?
The release.
The release kinetics.
Yes.
So we need to look into more in endosomal release, but it appears to be, so we looked at translatability of these RNAs that were introduced into the cells. So we use GFP, and we can see them being efficiently translated when compared to other lipid methodologies.
But it's still an interesting question
Yes.
to check the
Yes.
release kinetics.
So we do not have that information yet, but we do need to look closely at the endosomal release of these particles. And that will probably allow us to even modify them accordingly. So that's a very good point. Yeah.
Ivone, that was a great talk. Thank you. With respect to the codon optimization strategy, and without giving away any details of your strategy, how long do you think you can increase RNA stability within a cell, generally speaking? I guess maybe first start telling us, what is the normal range for RNA, the least stable RNA to the most stable RNA in a cell? What is generally that range, and how much do you think you can increase that?
If you notice the data that was presented, it's minutes. So RNA, the stability of the RNA, the more stable RNA would probably be hours. 4 hours, I will say, probably is one of the maximum that you will see. And you will think, how can you achieve a therapeutic benefit with that short time in which the RNA is present? There's pros and cons of having a shorter stability. And the RNA, again, the stability also depends on the protein that you're being expressed in, so depending on how stable your protein is. But you could probably double, we're expecting to at least be able to double the stability of the RNAs. In terms of stability, we're also looking at, we recognize that these sequences may allow the cell to recognize these RNAs as natural RNAs as opposed to foreign RNAs. So the cell has mechanisms to recognize RNAs as being attacked by viruses, especially if it doesn't recognize it as self. And so these could potentially result not just on higher stability, but also on less immunogenic molecules. So we're hoping to be able to at least double the stability of these RNAs. Thank you so much.
Thank you. Thank you for this great talk. And our next presenter is Dr. Gu. Dr. Gu received his Ph.D. in genetics from China, in China. Soon after that, he joined Dr. Fang's lab. In Dr. Fang's lab, he mainly focused on understanding the mechanistics and translational questions related to hematopoietic stem cells, specification and blood vessel growth during development and in disease. He has made great progress through his hard work and his good skill. Today he will present a paper entitled Zika virus protection by a single low-dose nucleoside-modified mRNA vaccination.
Good morning, everyone. Today I will present a Nature paper published this February. So for this journal club, the title is Zika virus protection by a single low-dose nucleoside-modified mRNA vaccination. So where has Zika been found? Before 2015, Zika outbreaks occurred in Africa, Southeast Asia, and the Pacific Islands. Currently, outbreaks are occurring in many countries and territories. So what's Zika? Zika virus is spread to people primarily through the bite of an infected mosquito. Zika can be passed from a pregnant woman to her fetus. Infection during pregnancy can cause certain birth defects. Many people infected with Zika virus won't have symptoms or will only have mild symptoms. Currently, there is no vaccine or medicine for Zika. So simply search the keywords Zika virus in Web of Science. Only several papers published before 2008, and there is no paper published during 5 years from 2009 to 2013. And after the outbreaks of the Zika virus in 2015, we can say there are more than 1,400 papers published just in 2016. And till now, there are already 400 papers published this year. So this data indicates that Zika virus is a very hot topic nowadays. Recently, several vaccine candidates for Zika have been developed, including plasmid DNA, purified inactivated virus, protein subunit, and adenovirus vectors. Compared to these candidate vaccines, mRNA has emerged as a promising vaccine and offers several advantages, including, first, its non-integrating and non-infectious gene vector that can be readily designed to express any protein with high efficiency, and its cost-effective and high scalable manufacturing. And third, its small dose can sufficient to induce protective immune response. First, in this paper, the authors designed a potent anti-Zika mRNA construct, concluding a singular peptide and the Zika premembrane and envelope glycoproteins, including of 5-terminal caps and 3-terminal UTR, like the poly-A tail. Then the nucleoside-modified Zika mRNA was prepared for vaccination by encapsulation in lipid nanoparticles. Zika mRNA was first characterized by transfecting HEK2-3 T cells and also human and mouse dendritic cells. The Zika envelope protein was produced by all cell type and was secreted into the superlint of HEK2-3 T cells. Others hypothesis that dendritic cells also secrete E protein and that it is rapidly internalized by endocytosis. The E protein in the superlint of HEK2-3 cells was penetrated by ultracentrifugation when incubated with PBS, but not with the Triton X100. Next, the immune response induced by the Zika mRNA vaccine was first analyzed in 57 BL6 mice. Animals were immunized by intradermal injection of 30 microgram of Zika mRNA or poly-C as a negative control. The INF gamma, TNF alpha, and the IL-2 production were stimulated with Zika E protein at week 2 after vaccination. This result suggested that Zika mRNA vaccination can induce the immune response in mice. The Zika E protein specific IgG in the serum developed quickly in vaccinated mice and stabilized at the endpoint titer of 180,000 at weeks 8 from 8 to 12. And the others used the two different independent assays to measure the anti-Zika neutralizing antibodies in the animals. The mean PRNT titer against the Zika virus peaked at around 1,300 and week 8 and was relatively stable until week 12. The mean RVP neutralizing antibody titer against the Zika reached about 100,000 at week 8 and 12. These two methods gave two different values but this difference has been reported previously. Next, the Zika mRNA vaccine was evaluated in the BA-ARBC mice. E protein-specific IgG in the serum peaked at week 8 and remained stable between week 8 and 20. PRNT neutralizing antibody increased to about 1,100 at week 16 and remained stable until week 20. The RVP neutralizing antibody titer reached about 50,000 at week 8 and remained about 20,000 until week 20. Next, a challenge study was conducted in BA-ARBC mice immunized by intradermal injection of 30 micrograms of Zika mRNA or policy controlled. Mice were then challenged by injecting Zika virus at week 2 or the short-term or week 20 or the long-term study after immunization. In the short-term protection study, control mice showed very near by this ray, and all the Zika mRNA immunized mice were protected against detectable viremia. In the long-term study, all the control mice showed viremia on day 3, while none of the Zika mRNA protected mice had detectable viremia at day 3 or day 7. This data demonstrated that a single immunization with Zika mRNA vaccine rapidly elicits durable protection against detectable viremia in mice. The authors next evaluated the efficiency of the Zika mRNA vaccine in the rhesus macaques. Macaques were immunized by intradermal injection with dose of 600 microgram or 200 microgram or 50 microgram of Zika mRNA vaccine. The E protein-specific IgG and the neutralizing antibody were efficiently induced by all 3 vaccine doses with no significant difference between groups. The absence of significant dose-dependent effect in the antibody response in any acid suggests that a low dose of 50 microgram was sufficient or possibly more than sufficient to induce robust anti-Zika immunity in macaques. Last, rhesus macaques were challenged at week 5 in 5 vaccinated animals and 6 control animals. All control animals become infected. By contrast, vaccinated macaques were highly protected against Zika infection. Summarization. In this paper, authors mainly did 2 kinds of experiments. The first kind is to test the immunogenicity of the Zika mRNA vaccine in different animal models. And the second part, they do the challenge study in these 2 animal models. And in these 3 types of animals, the authors mainly performed 2 kinds of experiments. One is to test the immune response by measure the E protein-specific IgG production. And another is to measure the neutralizing antibody by using 2 independent assays. Then the author get the conclusion. Nucleoside-modified mRNA lipid nanoparticles elicits rapid and durable protective immunity, and therefore represents a new and promising vaccine candidate for global fight against Zika. So this is all for this paper. Thank you.
Any questions?
Hi, Qilin. Is the RNA within the lipid nanoparticle being taken up by dendritic cells? Is that the mechanism? And the dendritic cells are displaying the antigen
Yes.
to the T cells, educating the T cells. Is that the mechanism?
They mentioned it, but they mentioned that this mechanism already published in the HIV virus. So they don't tell the details, but they mentioned that it's already been reported in other particles.
Thank you.
I just have a comment. This is a very nice paper for a journal club discussion. I think you guys selected a good paper. And because it gave us an idea of how mRNA can be used as vaccines and how we can adapt this for mRNA therapeutics as well. And what I wanted to see more in the paper was about the nanoparticles, how they use the nanoparticle delivery to both mice and monkey. Did they say more about?
No, they didn't mention it. So for this part, they should cooperate with the other labs. For their lab, they only focus on the mRNA vaccination, and not others. And also in this paper, they collaborated with another very famous lab. So that lab published one paper in 2016 in Science to use the DNA plasmid to make the Zika vaccine. But maybe the reviewers asked about they to compare the two different DNA plasmid and the mRNA, the efficiency. So they asked that lab use the DNA plasmid to do the same, to perform the same assay in the same lab and by the same stuff. So they get the same conclusion. Very interesting paper.
One thing I find interesting is that they are modulating the immunogenicity of the RNA. That's why they're modifying it to achieve an optimal response. So one will think that an RNA with highly immunogenic would be better for an appropriate response. But indeed, what they make a comment, no, that an optimized RNA is good for enhanced expression as well. But it's interesting that they use a modified RNA as opposed to a non-modified RNA. One thing, I mean, being from Puerto Rico, one thing I noticed also, they talk about, can you tell us more about, they talk about Zika versus dengue. So they're very similar. I mean, they're very similar, but they were able to find a domain that could confer a immunogenicity to Zika without causing problems with individuals that get affected with dengue, exacerbating a response to a dengue infection. Actually, they said that you could even have a stronger infection with dengue in some instances. So that's something to keep in mind when you're looking into developing vaccines. It's really, really good.
So I can have a follow-up question. So in Dr. Bonner's research, they actually modified the RNA and first injected T cell, and then this T cell kind of injected to the patient. But in this paper, it seems like the modified RNA is directly injected to the mouse. So what's the difference? So how can you ensure that this modified RNA can go to the target cell it's supposed to?
So for the T cells, in my opinion, it's targeted therapy because the T cells can recognize these special cells and or tissues. But in this paper, they use the nanoparticles, and this is non-specific target.
So it's presentation as opposed to targeting. Well, you have the T cell targeting, but they're using the antigen presenting cells to present the antigens to T cell and then confer long-term immunity. But it's interesting that they were able to achieve such a long-term immune response. I mean, they look at the response all the way to, how far did they go?
20 weeks.
20 weeks. So that's interesting. Did they look past 20 weeks? No, the maximum was 20 weeks.
So I think, Sahana asked a very good question and a little bit about the nanoparticles. She wanted to know a little bit more about the nanoparticles. And there might be some ways to enhance nanoparticle uptake by dendritic cells so that you get the antigen displayed on the surface of the dendritic cell to educate the T cells. So we're working on those things without disclosing any details, because it's something that we're quite interested in looking at. And we have different options here, as Ivone was pointing out. We have silicon nanovectors, we have lipid nanovectors, and now it's very interesting one, rosette rings. And we interesting to try those different nanoparticles in terms of their ability to be taken up by dendritic cells and provide an effective vaccine. One last question that might be on everyone's mind is, why are we, as a cardiovascular center, interested in vaccines, infectious disease vaccines? Well, we have a tool, right? We have mRNA therapeutics, and we're the only academic center in the United States that can make clinical-grade RNA. So we've got a hammer, and it's a great tool that we have, and we will apply it to cardiovascular. But we're still in the learning stages, and we're looking at low-hanging fruit. And other people that are getting into RNA therapeutics are looking at this low-hanging fruit as well. So vaccines actually have a place in cardiovascular disease. As Longo can tell you, or Henry Ponoff, who is here, I mean, vaccines are now being used for hyperlipidemia, as a treatment for hyperlipidemia. So we'll be able to apply our knowledge that we're gaining in RNA therapeutics to cardiovascular ultimately.
Okay, let's thank you again for both presenters. Great presentation.
We went